my great pleasure to be here to present my talk on personal exposometers for monitoring the environment. There's an outline of the talk. It's just overview of the exposome, the exposometers, and the exposome research, monitoring exposures at HEAP, and the summary. So I believe the entire audience is pretty familiar with the term exposome. And as Texas mentioned earlier, it is brought up by Christopher Wilde in 2005. And by definition, it covers a wide variety of factors that we encounter every day, ranging from the stuff that we can feel and touch, like water, air, and soil, and then to lifestyle, diet, and to ecosystem and social economic factors. We are particularly interested in the airborne exposome. So we are focusing on two parts of the exposome, the biological one that contains all the bacteria, fungi, viruses, animals, and the chemical exposome, which contains the pesticides, carcinogens, paints, and all the other nasty stuff that you could think of. Why do we study the exposome? The answer is pretty simple. The external factors interact with our internal environment and affect our health. The most well-studied airborne component will be the particulate matter. Those are small droplets in the air and can be categorized into groups such as PM10 that defines particles that are smaller than 10 microns, and the PM 2.5 for particles that are smaller than 2.5 microns. Once inhaled, those particulates can deposit into different parts of the lung and affect different health conditions. So both short-term and long-term exposures to those particulate matter can cause adverse health effects, not only in the respiratory systems, cardiovascular systems, and for the ultrafine particles, they can even go through the olfactory nerve and affect brain functions. Traditionally, those particulate matters are studied using the monitor stations. Those are shed-like structures outdoors and gives you a total concentration of particulate matter, a handful of gases, and some meteorological parameters such as temperature and humidity. There are many drawbacks associated with this approach. First one being low resolution. This is a fixed weather station, so there's usually only one in a large geographic area. A weather station providing data 20 miles from me is not gonna precisely reflect my personal exposures. Also, I spend most of my time indoors. The outdoor facilities does not accurately reflect my exposures. The next one is low information. It only tells me the total concentration of those particulate matter, but never tells me what exactly is in there, whether they are fungi, bacteria, pollens, or carcinogens. So to this end, we developed this personal exposometer to profile the exposome, both biological and chemical. So the top row shows some of the exposometers we have been using now, and we are prototyping. So what's unique about those exposometers is they are active sampling device. That means they have either a pump or fan inside of the device that's continuously drawing air, and there's a collection matrix to collect the airborne materials. For our exposometer, we have two collection matrices. One is for collecting the biologicals from the air, and this part will be used for sequencing. And the other part we have is a zeolite absorbance to collect the chemicals from the air, and then used for mass spectrometry analysis. So what can we do with these exposometers? In a previous study, we used the exposometer. Oh, this didn't show up. Anyway, we used. I'm so sorry for this. Anyway, it's just dots for the samples we collected from our participants. So we used our exposometers to track 17 individuals for 2.5 years. And this is what we found. So with our approach, we can detect over 2,500 species covering all domains of life, including bacteria, fungi, animals, plants, and viruses. This is a closer look at the composition of exposome. The top row shows the exposome composition across each sample period. Each bar represents one sample, and the color represents the taxons. The amber color is fungi, the red one is bacteria, the green is plants, and the blue one is mitozoa. So if you just look at the top row, you will see different colors popping up, showing this is a very diverse exposome composition. And if we cluster the exposome composition by the dominant taxon, we can see some of those days are amber colored, so they are fungi dominated days, and some are red, that's a bacteria dominated days, and the green ones are plant dominated days, and some of the days are mixed. So we could see that the composition of the exposome is very diverse and dynamic. We next want to investigate what's causing the variance in all those exposome compositions. So if we plot all the samples from P1 based on their location, we could see the samples from the same location clustered together. And if we do this for the season, we could see there are different clusters for each season as well. We do a variation partitional analysis, and that it reviews that the lifestyle and environmental factors are the top two that affect the exposome composition. So in a sub-study where we monitor 4 individuals across 3 weeks, all of these individuals live in the general San Francisco Bay Area. So the P6 is the dark blue dot, mainly lives in the San Francisco city. And the P3 commute regularly from East Bay to East Palo Alto. That's the the blue dot P3 across the bay. And P1 travels a lot. And during this sample period, P1 also traveled to Washington, D.C. And the P5 mainly reside in the South Bay area. If we plot all the samples from the individuals, we could see the samples from the same individuals clustered together, indicating a personal signature on the exposome composition. We could see, even though they all live in a general geographic area, their samples are still different from each other. We could see the nice distinction between P3, P5, and P6. For P1, who travels all around, his samples is more scattered around. So the location plays a big role in the exposome composition. It's quite intuitive to see that seasonal will have an effect on the exposome composition. Trees and plants blossom at different time of the year. And with our analysis, we also spot 4 patterns for our plants. For the exposome, we see some of those genuses that will peak at the spring and early summer, such as the pine tree. And the later one is false turkey tail. It's a fungi that peaks around the winter time. So season is another huge player in the exposome composition. What about the chemicals? On the chemicals, we have a similar picture. We can detect over 3,000 features from the chemicals that we collected. And the top categories include the pesticides, personal care products, plasticizers, and also carcinogens. We also spot some interesting effects of season in chemicals as well. Here shows 2 chemicals that shows different peak times across season. The top one peak during spring, while the later one that's mainly peak during winter. And we also found some ubiquitously found chemicals that includes plastic. That's basically everywhere. We found in every sample. And also pesticides that's also ubiquitously found in our samples. There are also correlations among our chemicals. The geosmin is earthy smell after rain. And the pesticides we found are in a reverse relationship with the plastic components. At 3 time points shown here, we see the geosmin and pesticides are at higher concentration, where the plastic components are at lower concentration. We think this could be related to the actual raining during those days. The earthy smells and the pesticides are actually brought up by the raining, while the plastic components are settled during the raining. Beyond the correlations between the chemical itself, the chemical components also correlate with biologicals. So here shows an example. Purine is a chemical, it's a carcinogen, and it's also commonly found in paints. We find a reverse correlation with the fungal species. Whenever we find purine, there's less fungal exposures. So if you are having a fungal exposure at your home, consider changing your paint, but also mind the concentration you want to use with the purine, because it's a double-edged sword. So how does the exposome affect health? Previously, I've shown that we have discovered thousands of stuff from our chemical and biological exposome. Then how does this affect health? So in this study, where we monitor one individual for 52 days using the exposometer to collect the external environmental factors, and we also collect stools and blood for gut microbiome, metabolome, proteome, and clinical tested blood tests as a marker for internal omics and health. So we did a spearmint correlation between the external environment with the internal omics. We could find almost 9,000 significant correlations between the exposome and the multi-omics. So on the right shows the correlation of those subdomains. The orange one refers to the chemical exposome. The purple bar is biological exposome, and the blue bar is the environmental data we pulled from a nearby weather station. On the right shows the different omics. The green one is the one that has most correlations. It's the metabolomics. The brown one is the gut microbiome. The red one is proteomics. And we also have cytokines and routine blood tests. So here shows the correlations between the exposome and the gut microbiome. On the left shows the correlation of the top gut microbiome and exposome components. The inner ring shows the gut microbes, and the outer ring shows the external exposome components. Again, the purple represents the biological components, and the orange represents chemical exposome components. We find over 1,000 correlations between the external environment and the gut microbiome. And we found some interesting patterns as well. For example, the Alistipis, which is involved in many inflammation and various diseases. We found this genus to be only positive correlated with chemical exposome components, but negatively correlated with biological exposome components. Rospira, which is thought to be one of the protective microbes, have a completely reversed relationship. It only negatively correlates with the chemical components of the exposome, but positively correlated with biological exposome. So this is a very interesting finding from our research. This indicates even though exposing to the same sort of external factors, this could lead to different effects based on the microbes, and this could change the internal gut environment. And the gut microbiome is not the only one that has correlations with our external exposome. In another study, we monitored the exposome, the skin microbiome, oral, stool, and nasal microbiome from 2 individuals, which we found that the skin has the most correlations with the external exposome, followed by the other 3 exposome. So this is makes sense, as the skin is one of the first layers that in contact with our external environment. The metabolomics is the omics that has the most extensive correlations with the external environment. Here shows the correlations between the internal metabolomics with the external exposome. The inner green dots shows the metabolites from inside the body, and the outside ring shows the exposome components. A red line means positive correlation, and a blue line means a negative correlation. We could see there are over 4,000 correlations, and they are mainly positive correlations between the external environment with the metabolites. We also did a pathway analysis. We found mostly there are the uh, amino acid metabolism that's correlated with the external environment. We do find some interesting patterns with the chemical exposome though. Some of the chemicals are only correlate with the metabolomics in one direction, meaning they only positively or negatively correlates with the signaling pathways. However, for biological exposures, they are both they correlate with both directions. Can be either um, positive or negative depending on the signaling pathway involved. So more importantly, we want to see how the exposome correlates with health. The omic profile is definitely gives us a lot of data. However, there's no standard comparing what is a good omic and what is a bad omic. So here we use the routinely used blood test as a marker for health. So this includes tests such as glucose levels, eosinophil counts, platelets counts, urine nitrogen, all this routine stuff you could see in the clinic. And we correlate external environment with the results from the blood test. We could see that for a lot of these biomarkers, the changes in those biomarkers are actually explained by exposome components alone uh, for the top ones that are inside the box. So the lower panel shows the contribution of exposome components. So the left one shows those markers are mostly related to the exposome components. On the right shows an example of glucose. So there are 5 glucose measurements during the study period, and the green belt shows the normal range of glucose levels. The bottom half shows the exposome components that are correlated with the changes in glucose. The chemicals are shown in orange, biologicals are shown in purple. And the leftmost box shows the direction of the correlation. A blue box means negatively correlated, and the red box shows to be positively correlated. We do see salicylic acid is negatively correlated with glucose. So this one has been shown in animal models to reduce blood glucose levels. But a lot of these findings remain to be tested, whether it's a true correlation or causality in vitro models. So the exposome research is great. Using the exposometers, we can profile lots of environmental exposures and correlate with internal omics. What about some challenges using those exposometers? Number one is a low biomass. So this the stuff we collect from the air is very low biomass. So that means if we want to do sequencing, the samples will be used for amplification. And as you all know that with amplification, you should introduce some bias for the microbes. And also because of this low biomass, it's super easy to get contaminated with the environment. And number 2 of the challenges is the limited real-time data. So with all the fascinating analysis we've done before, that's all offline analysis. It takes weeks to months to get the results. But for a regular user or study participant holding the exposometer, there's really no actionable insight at the moment. And there's a lack of standards. There's no standardized protocols of collecting, processing, or analyzing the exposome components. And there's no standards to compare whether this is a standard urban exposome or this is standard farm exposome. We have no idea. And for the databases, we use multiple databases just to analyze our data, and it's all spread out into different databases. But the good news is that the field is working together to get a more incorporated and inclusive database that we can find one to use instead of five or five or six databases to use just to understand the exposures. And the last one, but not least, is the correlation of causality. This is a quite difficult stuff to tackle with this kind of research. We found tons of correlations with the external environmental factors and the health, but are they really causal? There are lots of research to be done to demonstrate the causability of these correlations. So for the exposometer itself, this shows the exposometer we've been used prototyping and hope for the future. So right now the exposometers are powerful, but have some drawbacks. They are super costly. For this price, it only shows the exposometer price itself. It's not including the price for analysis. So itself can cost at least $600 just for the device. And it contains a lower flow rate compared to human breathing rate. So when you do a quantitative analysis, you want something that's closer to human breathing rate to mimic the actual exposures. And some of those devices have real-time PM2.5, PM10, which is the most well-known environmental factors. And we found that the regular population reacts much better to a number from the PM2.5. If PM2.5 equals 100, we know this means bad. We need to change to a different environment. But only some of these exposometer has this real-time data, and it's only a limited set of the sensor that's incorporated into the exposometer. Of course, in the future, ideal version of the exposometers, we want to have not only have the gas sensor to report the VOCs we encounter in our lives, but also can have the sensors for radiation, noise, UV radiation, and also incorporates a microneedle patch on your skin that can take a sample and gives out the result to you see whether the external factors are causing an internal response on your body. So what we do at HEAP, so at HEAP, we have a cohort of 100 women, either pregnant or non-pregnant. We profile these women using the exposometers I showed before. And we profile these women at 2 different seasons to account for the seasonal effects. We collect samples for both summer months and winter months. Some study design considerations. The final design is always a compromise between the participant and the study team. And with the exposometer use itself, it comes with some certain restraints. From a participant point of view, they care about the sample duration and frequency. So a one-time sampling is much easier to commit to than a longitudinal profiling. And with the exposometer itself, can it be placed at home, or do they have to be wear on the person all the time? If it has to be wear on the person all the time, how often does it need to be charged? Does it look discreet? Can I just put it in my purse and carry it all day? Or do I have to put it on a visible position? Then what about others? Will they think I'm weird if I'm carrying this exposometer with me? Do they think I'm seriously sick? And what about the noise? The exposometer is an active sampling device with a pump inside. So there's a noise coming from the pump working itself. So is it a loud noise? Is it gonna disrupt me, my work, my sleep? What about others? If I put that on the tray table inside of an airplane, will my neighbors complain and just throw away my exposometer? What about ease of sample collection? Do I need to go somewhere special to get the sample collected? How about the data feedback? When do I get the data feedback? Are there any actionable insights from those data? The participants not only want to contribute to science, but they also want to make their lives better by participating in these kind of scientific research. So from the study team point of view, the amount of sample collected is definitely a huge factor. We always want more samples. and we want more collected from those samples because it's low biomass. If we collect for a relative longer period, this got more samples collected, and this less prone to other factors that contaminates our samples. And longitudinal sampling is always better than a 2 time point sampling, especially when we're looking at the exposome components. Also, offline analysis gives us the most detailed data of the exposome, but sure, it will take a long time before the data delivery. So in the end, with our study, we collect exposome samples every 2 weeks and at different seasons to account for all these effects. So our participants will wear one of these UPass exposometers shown on the left. The very left shows the filter cartridge open. From top to down, you will see a disc filter that looks like paper. Those are the ones that we use to collect the biological components, and which will then be used to extract DNA and RNA and then subject to sequencing. And the lower one shows that looks like a bag of sand. That's actually a zeolite absorbance beads that collects the chemicals from the air, and then we use that for mass spec analysis. So on the chemical side, we get the zeolite beads from the pouch that we have inside of the exposometer, extract the chemicals using methanol, and then go through the mass spectrometry and data analysis using the XCMS pipeline. There's an example of the QA/QC we've done with the chemical exposome. We see the QC groups are clustered together away from samples. This means this is a good run. And for the also data pre-processing, we replace missing values, we normalize our data and do log transformation, and also Pareto scaling. On the biological side, on the sample process, the middle one shows a use filter from the participants. You will see a darker circle in the middle, and then the outer circle is less used. So to avoid contamination, we process all those samples inside a biosafety cabinet. Also, all the pre-amplification period processings are separated from the post-amplification. So after those samples are amplified, we take it out of the hood and then prepare the sequence library. We use this BioMac from Beckman Coulter. It's an automatic platform that allows us to process 96 samples at a time, and the output from this BioMac platform is quite even, and we can see from the right it's a bioanalyzer trace from the samples that we used. It's very uniform across all samples, and the fragments are the ones that we want to use. For the sequencing platform, we actually use a Ultima sequencing platform. This is a rather new platform. So similar as Illumina sequencing platform, this is also sequencing by synthesis, but the difference is that this is the incorporation of the base is reversible, meaning those bases can be washed out at the next step, and this makes the entire sequencing platform a lot more faster and low cost. For the analysis pipeline, we have our raw reads going through the QC steps, remove the human reads from all those results, and then do a de novo assembly into assembled context. And then the context are queried against our own custom-made database to allow us to do the taxonomy downwards. So for our part, there are lots more data to count that includes the exposome profile patterns, the personal pattern, the longitudinal pattern, and also the seasonal and locational patterns, because our cohorts coming from different cities of Finland, and also the difference between pregnancy versus non-pregnant women. Also, there's more importantly, want to see the correlation between the exposome with the metabolomics. We have samples collected from urine, And we plan to do a metabolomics with those samples to correlate the external environment with the internal environment. And for the correlations with offspring health, this will be for future project to see how the exposures during pregnancy will affect the offspring health. And with that, I'd like to uh, express my thanks to Joachim Dillner. It's my great pleasure to participate in this project. And also I'd like to thank our HEAP partners, Wiley, especially for handling the entire cohort, and also our study participants, which are amazing, and there are only a few of them have dropped off from our study, which never happened in all the other studies where we have demanding the demanding characteristics. Also, I'd like to thank Mike Snyder and also our colleagues from the Standard Lab. Last but not least, I want to thank the European Union for the funding, and thank you all for listening.